Hi, good afternoon. Thanks very much for joining us for another weekly research conference. Bonjour, bienvenue. And this is a very special, exciting session featuring a fantastic speaker with cutting edge science. And now before introducing our esteemed speaker, just a few logistics, which I think are pretty familiar for folks now, that include the fact that if you have questions, we very much welcome you to pose the questions as the lecture is in progress using the Q&A feature on the bottom of your screen. And we'll endeavor to answer them at the end of the presentation. And also, if you want to ask the questions live, please also do so by using the hands up feature, and we'll unmute you and invite you to join in on the conversation. And but if you have any technical difficulties, please use the chat feature, and Kelsey or Allison or our team member will help you to resolve the technical issues and get you linked in. Now, we're delighted today. We have Dr. Katey Rayner, one of the foremost vascular biologists, to give us a latest update in terms of her research. And folks know that Katey is a scientist and director of the Cardiometabolic microRNA laboratory here at the University of Ottawa Heart Institute. She's also associate professor, Department of Biochemistry, Microbiology, Immunology at the Faculty of Medicine, University of Ottawa. And she actually will be taking on greater responsibilities at the Faculty of Medicine. And she leads the Ottawa Regional Strategic Research Hub on Vascular Inflammation and Atherosclerosis. She obtained her BSc originally at the University of Toronto and then subsequently PhD at University of Ottawa. Her doctoral work focused on the role of heat shock proteins and macrophage foam cells in the development of atherosclerosis. She then followed with a postdoctoral fellowship at the Harvard Mass General Hospital and the subsequent New York University with Dr. Catherine Moore, where she identified the role of microRNAs in the regulation of HDL and the processes involving atherosclerosis, and for example, specific roles of microRNA 33 and the atheroprotective effects. Then we're very happy that she returned to the Heart Institute, and her research program focused on how inflammation is dysregulated both in atherosclerotic vascular disease and other metabolic diseases that's relevant in terms of progression of cardiovascular disease. And she particularly focused on also area of intersection of inflammation, energy metabolism, RNA biology, and also translational research on, for example, how microRNAs can be used as therapeutics. More recently, she also pursued the role, for example, of a necrotic factor RIP kinase 1 in obesity and atherosclerosis, and gathered a lot of momentum and attention of this work. Now, these series of investigations led to high impact publications, journals, including Science, Nature Metabolism, Circulation, Circ Research, JCI, amongst many others. And she's a member of peer review committee for the CIHR, the Heart and Stroke Foundation, and the National Institute of Health. She's on the editorial board of the journal Atherosclerosis Thrombosis Vascular Biology, or ATVB, as well as Circ Research. And she is the social media editor of ATVB, and we get regular postings, announcements, from Katey. And in recognition of her research excellence, she's been bestowed the New Investigator Award from the CIHR, McDonald Scholarship from the Heart Stroke Foundation, and her work has been funded by CIHR, the Ministry of Research and Innovation, and European ERA-CVD Consortium, which is really exciting, looking at the vascular impact on brain and the heart. And she's been recognized by American Heart Association's award, including the Irving Page Young Investigator Award and the Early Career Award for Outstanding Research. But most recently, she received the prestigious Joe Vita Award from the AHA, recognizing really highly impactful work and innovation in cardiovascular biology in the last 5 years. So really congratulations. So today we look forward to her presentation. It's entitled Cholesterol, Inflammation, and Atherosclerosis, Expecting the Unexpected. Okay. really looking forward to the unexpected part. Yes.
Thank you, Peter. Yeah. Let me just share my screen here and you can let me know if you can see it okay. Is that all right?
Looks good, yeah.
Okay, great. All right, so thank you, Peter, for that very, very lovely introduction. So yes, as Peter mentioned, the title of my talk is basically Expect the Unexpected with regards to cholesterol and inflammation. And what I thought I would do is sort of give an overview, a little bit of a state of the art in terms of atherosclerosis and sort of the concepts that we're all talking about now. And then of course, tell you about the work that we're doing in my lab in relation to this. But the reason is that this field is changing rapidly, and the concepts that we're realizing contribute to disease is also changing rapidly. So there needs to be a bit of an update with regards to these mechanisms to frame a lot of what we're doing in my lab. So always starting with the ending to really acknowledge the people in the lab that do the work. As you can imagine, this is all an amazing feat for our team to do all these really cool projects that I'm going to be telling you about today. So I just want to highlight Michelle, who's in the middle here. She's our lab manager. She keeps us all in line. Adele, a postdoc in the lab, whose work I'll be talking a little bit about today. Cameron, who's a master's student, hopefully soon to be PhD student in the lab, whose work I'm also going to be highlighting today. And Leah Susser, who's a PhD student. And of course, the rest of the team is all here and working on, either individual projects or supporting the ones that I'm going to be talking about. But I really wanted to highlight these individuals because they really just make coming to the lab really, really fun. And they're all so motivated and so talented. So the reason that I get to have my name on those awards is 100% because of them. So as I said, thinking about the disease of atherosclerosis, of course, we know it develops over many decades. It's the disease that causes coronary blockages that eventually lead to MI. This happens again over a long period of time. Risk factors are well known, including LDL cholesterol, smoking, lifestyle factors, sedentary lifestyle, et cetera. Hypertension and age and genetics. So a lot of these, genetics contributes to a lot of those things. So, genetics is not entirely separate from the also the modifiable risk factors. And of course, these are LDL cholesterol and hypertension in particular are the #1 ways that we treat this disease. But really, atherosclerosis in the coronaries and elsewhere actually is really a chronic inflammatory disease. So it's in reaction to the cholesterol that is elevated and it gets trapped in the vessel wall, but ultimately it is a chronic inflammatory condition. So instead of just thinking about, these traditional drawings that we have where these yellow streaks, these fatty depositions, we really have to think of cardiovascular disease, atherosclerosis, and the subsequent MI that happens as an inflammatory reaction. All of that revolves around the immune cells that are produced often by the bone marrow and the spleen, and I'll talk about that a little bit, but that ultimately end up in the circulation where they can then go to the atherosclerotic plaque and they contribute to the growth of the actual plaque itself, but then they also go to the heart following an MI. That can lead to both the injury that the heart sustains after MI, which is causing a lot of that fibrotic and the malfunction in the myocardium, but it also leads to actually the repair as well. So understanding the immune system is really, really, really critical, and understanding the factors that control the immune cell production, whether it be in the circulation, in the bone marrow, in the heart itself, is also of utter importance to make sure we understand the full mechanisms of this disease. So atherosclerosis in the vessels, again, really is central. The central player in the development of atherosclerosis is the macrophage, and this is derived from monocytes that come from the circulation. And the macrophages then enter the plaque or the subintimal space right beneath the endothelium. And they do so in reaction to the excess cholesterol the LDL cholesterol in particular, that has accumulated in that subendothelial space. That cholesterol signals to the immune system that there's something wrong. The LDL particles get easily oxidized, they become aggregated. And that's a signal to the immune system, that something needs to be cleared up and cleared away. So the monocytes are recruited, or the intimal resident macrophages are activated, and then they gobble up all of that cholesterol and they become these foam cells, which are full of lipid. They can proliferate, they can signal to recruit more monocytes and macrophages into the area, and so on and so forth. And again, this is something that happens over many, many decades. It's not an acute event. It's not in relationship, to a specific injury or specific pathogen, of course, but it is, it mimics that process very much. And then from there, the balance of how the cells and how the plaque grow really has to do with how many more monocytes or macrophages are being recruited to the area, how they interact with the smooth muscle cells and the endothelial cells in the region, how they can either leave if they've been able to clear away the lipid and are able to egress and go back to the circulation and clear away the debris, or if they die, of course. So if they, you know, and an excessive amount of cell death happens, of course, we end up with a lot of necrotic debris, and necrosis and necrotic core, we know, is really a hallmark of a vulnerable plaque. So thinking about sort of these two ends of the spectrum then is sort of a lot of the focus of what my lab has been thinking about over the last kind of five years or so. And that is the production of those immune cells that lead to that initial accumulation and that reaction to the accumulated LDL, as well as, you know, what happens when these massive amounts of cholesterol accumulate in a cell and then the cell undergoes cell death, you know, what happens then? So starting with the cell death first, cell death really is divided often into two forms, programmed and non-programmed, or apoptosis and necrosis. Apoptosis involves caspases. It's, you know, an intricate and complex cell cascade that is very well controlled, as you can imagine. The cell ends up in this sort of clean, tidy bleb. It's recognized by local phagocytes and cleared away. And in fact, it's not only non-inflammatory and normal, but it's actually somewhat anti-inflammatory, and it can actually initiate tissue repair. Necrosis is the opposite. Most of the time, the cell is reacting to some sort of physical or chemical injury. The cell bursts. It releases all of its contents. Those contents signal to the immune system that there's been an injury. The immune system then reacts. It reacts and produces more pro-inflammatory cytokines, which recruit more immune cells, and the inflammation ensues until it can be cleaned up. But in fact, there's sort of a hybrid type of cell death, and that's programmed necrosis. So it's still very much cellular signaling regulated. It's very tightly regulated, but the end result is the same as necrosis. The cell bursts, the outside contents are released, and that signals to the immune system, you know, again, that there's been an injury. And the thoughts about why this type of cell death exists is because if a cell were to need to undergo apoptosis, let's say there was an infection or a mutation, but something was wrong with the apoptotic machinery, the caspases were inactive, maybe they were themselves mutated, the cell has sort of a backup. It can undergo cell death and still, you know, be eliminated from the host. And then it does incite inflammatory reaction because, you know, there is something wrong. So there is, you know, the thoughts of why this exists. But as you can imagine, there's lots of things that trigger programmed necrosis that are not, in fact, you know, tumors or infections. There's loads of other things, which I'll talk about in a second. And that type of cell death, that programmed necrosis, is called necroptosis. It's, again, a hybrid of these two words. So we've been studying this for a while in the atherosclerotic plaques. We've been doing this in mouse models and in humans. We started off using a small molecule inhibitor of the whole pathway, and we found that in mice in particular, you could really remove that, prevent that necrotic core from happening in the atherosclerotic plaque if you use this special inhibitor called NEC1. And you can just see here the acellularity in these mouse plaques. And when you treat with that necroptosis inhibitor, it's really much more cellular, and the plaques are much smaller. Others have done things like genetically ablate one of the key players, which is RIPK3, which is sort of part of the necroptosis pathway. And same thing, in mouse models, you really do see a reduction in the amount of plaque. More recently, we went a little bit upstream and we used an antisense inhibitor to another key player in this pathway, RIPK1. And we found that indeed, RIPK1 inhibition also really reduces the development of atherosclerosis in mice, and partially because some of this pathway actually crosstalks with NF-kappa B and inflammation. So it's not just the cell death that's contributing then to the atherosclerotic plaque and the growth, but actually the ability of this pathway to also tell immune cells to become activated and produce cytokines. So, you know, this pathway is really ripe for therapeutic intervention, not only because of the death part of it, which, as you know, as we can see in mice, really contributes to the necrotic core, but also from the inflammatory point of view, especially in endothelial cells, actually. Endothelial cells have a really, robust activation of NF-kappa B by this RIPK1. And so silencing it, we think can really, really reduce that activation in the artery wall. But one of the other players is called MLKL, and I'm not going to go into, it's a very complex signaling pathway, but I will tell you that it's this really interesting protein that oligomerizes, goes to the cell membrane and causes that rupture. So it's really MLKL that is that final step. and the one that is really responsible for the cell bursting and inducing the necrosis that is subsequently inflammatory. So we just hypothesized that if we block MLKL, because it's the, you know, that ultimate step that causes the cell death, likely we would reduce cell death as well as reduce atherosclerotic development in mice. And part of the reason that we guessed that was because when we looked at human coronary arteries, this is a coronary artery here with obviously a massive complex lesion with a very, very, very tiny lumen here. And when we looked in and around the necrotic core of these, or the necrotic regions of this artery, these little brown dots here are positive stains for this phosphorylated MLKL, this active MLKL. So we really saw that MLKL was present only in these advanced necrotic regions in humans. So this is the quantification here. So really, you know, when you looked at sort of normal arteries or just a little bit of pathological intimal thickening, we didn't see much of this terminal MLKL signal. So likely, you know, our hypothesis was then, you know, by definition of the way that this necrotic core maybe develops, maybe MLKL is really acting at that terminal step near the ages. So we thought maybe knocking that down and in mice would be able to prevent a lot of the subsequent inflammation that happens in atherosclerosis, like we'd seen with some of the other models. So this was work done by Adil in the lab, and he showed that in ApoE knockout mice, which is sort of a run-of-the-mill atherosclerotic model, indeed, if you take antisense oligonucleotides, these are just, you know, siRNAs, therapeutic siRNAs that you can just give subcutaneously, and you gave these mice a Western diet, you did see actually a reduction in the necrotic core, just like we thought. So if, you know, MLKL was indeed leading to the necrosis that we had previously observed, you know, we previously observed in humans. But when we quantified the size of the plaques, and usually when a necrotic core is reduced in a plaque, the plaque is also less advanced. Often that's because, you know, there's fewer cells, the cells don't get recruited, the death doesn't then lead to further recruitment of cells. But we saw something very curious. There seemed to never be, there definitely wasn't a reduction in the size of the plaques these mice had. They actually trended towards being elevated, larger than their control counterparts. And that didn't make any sense to us, given how we, you know, we're thinking about the way that necrosis happens and the inflammation that happens subsequently. But when we looked closer, what we noticed, just let me say that we confirmed that indeed the cells were not undergoing cell death. So we used the cell death stain tunnel and looked in the artery wall, and you can see in the control mice, you can see little green dots and indicating these cells are undergoing cell death. And there was very, very, very little cell death that was happening when you didn't have MLKL. So as we suspected, you know, when you inhibit MLKL, you don't get the same amount of cell death in the plaque as you normally would. But when we looked closer, we noticed this red stain here is a lipid stain. So it's showing all the lipids that have accumulated in the artery wall in these mice. And you could just see here right by the visual that there was more of this red lipid stain in the mice that didn't have MLKL. And when you quantified it here, it indeed showed that these lipids, actually, there was more of these lipids within these plaques. So the plaques were sort of better off because they hadn't undergone all this cell death, but they were worse off because they had all this extra lipid. So we went into a lot of this mechanism, which I'm not going to cover, but suffice to say that MLKL, you know, functions not only as a cell death initiator or cell death, executioner. It doesn't just go to the plasma membrane and cause cell rupture and necroptotic cell death. What others had shown, other types of cells, was that MLKL could actually help traffic these multivesicular cellular bodies in the cell, and it could actually enable the secretion of exosomal vesicles. So it wasn't just actually this, you know, terminal step of necroptosis, this protein. It was really, really important for the way that the cell actually traffics various things around the cell. What we found, and again, I'm not going to show you the data, but what we found is that under normal circumstances, we think what MLKL is doing is in a lipid-laden macrophage, like we see in the plaque, it is needed for that lipid to get in and around the cell and get out. And if you don't have it, although yes, you save the cell from not dying and you're able to sort of prevent that inflammation and necroptosis, you actually are not able to traffic those lipids out like they need to. So they're not able to undergo, their typical metabolic disruption and efflux out of the cell. So in fact, MLKL is clearly playing so much more of a role in the atherosclerotic plaque and in the cell itself beyond what we initially thought. And so this has led to a lot of different cool things regarding how MLKL is sort of this death protein that's way more than death protein. But I want to just highlight, kind of going back a little bit to this concept of that influx of immune cells that are, also contributing to the way that the plaque develops, because that sort of leads to the next thing that we found with regards to how MLKL might be acting in the atherosclerotic environment. And that's that, as we say, it turns out that the bone marrow, as we know, is a significant, is the main organ that produces the immune cells from their progenitors. And when the body needs it, they can produce, monocytes, T cells, B cells, whatever's needed. But at times when the body may need more immune cells, or if there's something wrong or the bone marrow environment has been compromised, the spleen can take over. So a number of years ago, Phil Swirsky, Matthias Nerendorf, and Clint Robbins had found that the spleen actually was a major place during development of atherosclerosis that actually had this reservoir of immune cells that then contributed to the development of the atherosclerotic plaque. So if you took out the spleen of mice, their plaques were smaller. So the bone marrow was fine, was intact, but their spleen actually contributes directly to these. So, understanding then the bone marrow and the spleen and how they produce these immune cells will then obviously lead to a better understanding of how, to prevent either the recruitment of monocytes directly to the plaque, or the way that the macrophages actually react once they get there. Because what Adele noted was that in these mice that didn't have that MLKL, where MLKL was inhibited, they had massive spleens. And for those who are not used to looking at mouse spleens, this is a normal mouse spleen. And this was one after MLKL had been blocked using these antisense inhibitors. And this spleens were absolutely huge. In fact, thankfully, this phenotype was something that we didn't have to go look for. It was staring us literally right in the face because it was certainly not something that we expected at all. It had nothing, we'd never heard of MLKL controlling anything regarding the spleen. There'd never been reports of that before. So it was something that, that was really completely impossible to miss. And when Adele looked microscopically, he could see that the red pulp to the white pulp ratio was much, much, much larger in mice that received that MLKL. So it wasn't just that the spleens were larger, but the actual red pulp, where a lot of those immune cells and the macrophages and the monocytes really expand. That particular environment, specifically in the spleen, was expanded. And when we flipped it a little bit and we asked, well, what about just during atherosclerosis in general? What does this spleen look like? What does the MLKL look like in the spleen during, in mice that are developing atherosclerosis? And if you have a mouse fed a chow spleen, this is the expression of MLKL. You can see it's very restricted to the red pulp. And when you put these mice on a Western diet, this expression of MLKL is really, really diminished. So it seems like MLKL is endogenously lost during this, progression of atherosclerosis in the spleen. And that's really important because that coincides with what had been shown previously in terms of this is when the monocyte production in the spleen that then leads to the macrophages that develop, that contribute to atherosclerosis. That's really what's happening is in the spleen, we get this expansion. So seeing the loss of MLKL really is mirroring what we're seeing when we inhibit MLKL using an antisense oligonucleotide. So then we asked, whether or not there's an effect of the loss of MLKL directly in the spleen. And again, I don't have time to go over all the data. Suffice it to say, it really does. MLKL seems to play a really, really important role in immune cells in the spleen and in the microenvironment in the spleen, even more importantly. So when we look and this is just some summary data here from some flow cytometry that Adele did on the spleens from these animals, we see a far greater number of progenitor cells, both the hematopoietic progenitors as well as myeloid and then mature monocytes in these spleens. So there's something very unique about these expanded spleens where they're really expanding their monocyte reservoirs, both all the way from progenitors all the way to the mature cells. When we take spleens out, you can actually culture spleen cells and splenocytes and see what colonies form as a reflection of the immune progenitors that are there and their propensity to become myeloid or lymphoid or other cell types. And when you don't have MLKL in the red bars, there's pretty much every type of colony that you see in the myeloid lineage is expanded in a culture dish as well. So this is mimicking what we see when we look directly in the animals. And again, I don't have time to go into the details here, but actually what we found is it's not the immune cells themselves that have any inherent, properties that are defective or tend to, want to become myeloid cells or more of them or proliferative. It's actually the environment that they're in. It's the splenic microenvironment that is deeply impacted by the loss of MLKL. And I'll touch on that again in a second. But we think that it's not, so here we are adding to this understanding of, immune cell production by the bone marrow and the spleen to say it's also not just the bone marrow and the spleen immune cells, but it's actually what they're surrounded in, the stroma and the vascular cells that are in there that are controlling immune cell function that contributes to disease. And this was shown really recently, just a couple of weeks ago, actually, in a lovely paper by Matthias Nehrendorf and colleagues. And what they found was that in patients with various risk factors for cardiovascular disease, so individuals with hypertension, atherosclerosis and hypertension, or myocardial infarction, when they looked at the bone marrow of these individuals, this is not the circulating cells, these are actual bone marrow of individuals. They looked at these progenitor cells, the immune progenitors that will eventually go on to become those immune cells that lead to plaque development. And there's an increase in the proliferation of those cells in these individuals with the various types of risk factors for cardiovascular disease, or having had an MI previously. So it sort of was proof of concept that in the human bone marrow, the immune cells are sort of already activated and already, becoming a little bit more primed. And they seem to be all related to one another. Hypertension, atherosclerosis, MI obviously are all highly related. When they could model this and measure this a little bit more closely in mice, they took mice and they did, a hypertension model, an atherosclerosis model, or an MI model, and they saw basically exactly the same thing, where they could really see an expansion of these immune cells. But what they noticed was in the vessels in the bone marrow. So here are the bone marrow blood vessels here with the endothelial cells here. They saw an expansion of the vasculature in the bone marrow in mice and with all of these different cardiovascular risk factors. And when you took the mice that were in the atherosclerotic environment, this red stain here is lipid. And that lipid, it's the same stain that we used in the mice that lacked MLKL in terms of seeing that expansion of lipid in the atherosclerotic plaque. And you can see here the quantification. So in the bone marrow, the endothelial cells accumulate lipid in the setting of atherosclerosis. They have a whole, this paper goes into some really cool mechanism, but the endothelium itself in the bone marrow is part of the way that the immune cells are then produced, which are then the ones, who go on to exacerbate atherosclerosis and MI. So now we're really realizing that it's really the environment that the cells are in, not just the immune cells themselves. And here's the various, a graphic of just the hypertension, atherosclerotic, MI. And, there's proliferation of the immune cells, increased monocytes, which then go back and feed, back to the artery. There's a question, of how these inflammatory cells get activated. There's some mechanism that they have figured out that it's related to IL-6, but we have evidence in the lab now that MLKL might be really key to these endothelial cells, in particularly the spleen. So if MLKL is there and present, we think the immune cell, the endothelial cells are able to control immune cell production quite easily, and they're able to sort of, maintain the expansion. But if MLKL is absent, the endothelial cells become dysfunctional. We're not sure why yet. And that enables this, expansion, this sort of uncontrolled expansion of immune cells that we're seeing in the spleen, which then, of course, eventually contributes to inflammation in the plaque. So in terms of, just other things in our environment that, activate monocytes and promote the monocyte production in the spleen and in the bone marrow that lead to the inflammation that we see in the heart. So stress is not so shockingly associated with an increased risk of cardiovascular disease. And in humans, you can see that on a perceived stress survey, these are ICU nurses, and this was before the pandemic, so who knows even now how much worse it might be. A perceived stress scale after when being on duty for ICU associates with an increased amount of leukocytes in the blood. So we know that stress can cause immune cells to enter the bloodstream from the bone marrow. And this is done in mice. So putting mice under stress, you see this increase in the amount of these progenitors in the bone marrow of these mice. This has just done some imaging that was developed here to show that, again, the stress, these are simply just mice under stress. There's an increase in this inflammation, this protease activity that they detect in the heart. And this is an atherosclerotic mouse, which has, developed larger atherosclerotic plaques, simply from being under stress. So, you know, it's linking a lot of these factors that we realize are risk factors for coronary artery disease, and it's linking it back to the way that the monocytes are affected. Now, we don't know yet if, you know, for example, in the case of stress, if this is related to the way the bone marrow microenvironment might sense stress, whether the immune cells themselves are just sort of inherently different and, you know, acutely versus long-term, you know, what the effects are. But, you know, certainly it's something that we're all thinking about. Sleep is another big one. So this is a very cool study where they took animals, they took mice, and they disrupted their sleep continuously during their sleep cycle. And so it's not a model of sleep deprivation. It is just a model of sleep fragmentation. So I think it's something that a lot of us can relate to. So complete sleep deprivation actually puts humans and animals into more of a stress response. But in this case, sleep fragmentation is more just a sort of a continuous disruption in sleep, but still the animals are able to get sleep. And you can see here in the green bars, this is just SF, means sleep fragmentation. The atherosclerotic plaques are much larger, simply from having fragmented sleep. And that is paralleled by increased monocytes in the blood and increased monocytes in the bone marrow. And the idea being that sleep fragmentation has a specific way of talking to the bone marrow to produce more immune cells. And that eventually is some of the reason why we see increased cardiovascular risk with individuals who have fragmented or disturbed sleep. On the good side, there's the same mechanisms are at play with things like exercise. So in mouse studies, if you allow animals to exercise sort of freely, turns out mice exercise continuously, virtually all night. And as they exercise, they have a reduction in the amount of bone marrow. This is bone marrow here. Bone marrow monocytes in their blood and in their spleen, pretty much everywhere. So all of the mice that are allowed to exercise continuously have overall a reduction in the circulating leukocytes. And again, unsurprisingly, this leads to a smaller lesion size in the animals that have been allowed to exercise. Now, this can be measured somewhat in humans. Again, we know that exercise, of course, contributes to a reduction in cardiovascular events, but it does correlate actually with a reduction in the overall amount of leukocytes that are in the blood. So this study was just done in the blood, but presumably, this is something that we could then extend and say, what is going on in the bone marrow, what is going on in the spleen, how do these immune cells actually behave and act differently if they're doing exercise versus sedentary. And so, these are, again, we're primarily talking about the traditional risk factors for atherosclerosis and MI and things like that. Cholesterol, things like lifestyle factors like sleep or stress. But of course, there's more and more appreciation for the role of other risk factors for MI, particularly infection. In post-COVID, of course, I think this will become a completely different story that is, made even worse, obviously, by the stresses on the cardiovascular system by COVID. But in this study a number of years ago by Vicente Corrales Medina here in Ottawa, showed that just having had community-acquired pneumonia over time, up to 10 years post-infection, actually ended up with up to a 1.5 to 2-fold increased risk of developing a subsequent cardiovascular event. So just having had, and this is matched for risk factors, so it's not that just over time or over age. So there's an increase in cardiovascular risk acutely, which can be sort of imagined by potentially acute inflammatory stress. But even over time, over potentially up to a decade, individuals who've had community-acquired pneumonia continue to have a risk of MI or other cardiovascular events. More recently, everybody here in Ottawa, including Vicente and others here at the Heart Institute, found if they used PET imaging, that just after discharge, after resolution of pneumonia, there was still persistent inflammation in the lungs of individuals up to, in this case, 45 days post-discharge. So there's something that persists in the inflammatory environment in the lungs many, many, many weeks after the initial infection. The infection has been cleared. There is no acute infection anymore. But certainly there's something related to the way that that inflammation is persisting that is leading to a potential increased risk. So what are those mechanisms? That's really interesting to us. So why is there something that, over many years potentially continues to put an individual at risk of developing an MI or similar? And of course, there's a ton of therapeutic opportunities you can imagine here in this space. So we can model pneumonia infection in mice, and this is work that Cameron is doing, Cameron Stotts in the lab. And it's not easy, but you can give mice pneumonia infection. And this is a good time to shout out to ACBS as well for their significant help in helping our mice get pneumonia. And then we have the ability to look then similarly at the atherosclerotic plaques and ask, does the inflammation that we see in the lung, does it match what we see in the plaques? Is it making it worse? What are the mechanisms? What are the things that are changed? And what are the things that are elevated? So this project is just getting off the ground, so we don't have much understanding of it yet. But Cameron has already, again, mastered the infection. We do see that, if the inoculated mice here have much higher inflammation in their lungs. So we are indeed able to, get this lung infection up and running. These mice recover. Their inflammation returns back to baseline, and they're able to recover. So we're now investigating what impacts that may have on the atherosclerotic plaque. But that's sort of, mechanisms and understanding. But in terms of therapeutics, we're also able to understand, what kind of therapeutics might we be able to develop that might be able to reduce that inflammation. And we have a collaboration with Suresh Getty at BMI, and he's developing nanoparticles. And if we throw a pro-resolving lipid mediator, EPA, or fish oil, also known as Vasepa, into nanoparticles, and we can actually treat cells in culture to understand the inflammation, as well as then eventually treat the mice, who've had this infection and who are atherosclerotic as well. We can better understand, how we might be able to develop a therapeutic. So we're able to really see that the nanoparticles that contain that EPA, which is that fish oil here, that we know has resolving properties. We're able to reduce the amount of IL-6 when we treat with a bacterial product, LTA. And these are some recently acquired images that we're really excited about. These are mouse lungs that have been excised and labeled with a fluorescent, when the nanoparticles that were delivered had a fluorescent tag on them. And we can see indeed that the nanoparticle delivery platform that we're developing indeed accumulates in the lungs of mice. So this is really exciting. Now we're able to understand not only can we treat some of this inflammation in a more targeted way in the lung, but of course, can we also treat the inflammation that is secondary to that in the heart and in the vessel wall? And so another quick little story here is just the idea that aging, as we know, is a major factor in the development of cardiovascular disease, but aging, maybe unsurprisingly, affects the bone marrow as well. And one of these really exciting emerging concepts is clonal hematopoiesis. And the idea is that as individuals age, some acquire somatic mutations in bone marrow progenitors. And those bone marrow progenitors with these specific mutations tend to gain a clonal advantage. So they outgrow over time, and they sort of compete out all the other myeloid cells in the bone marrow. And those particular mutations, here are some examples: TET2, DNMT3, and JAK2 have been shown to have, these are not leukemias, these are not blood cancers in any way. These are really just, when immune cells in the blood sort of all have the same properties, there's individual mutations in some or more of these genes that have led to an expansion of this particular sort of pool of monocytes. And it turns out that those monocytes then have increased production of things like IL-1 beta, IL-6. They tend to go to the plaque more through many different mechanisms. They tend to contribute directly to ischemic heart failure in the heart as well as increased thrombosis due to some various mechanisms involving NETs. So there's clearly something regarding, over time and age during these acquiring of these particular mutations that leads to the inflammation that these monocytes and macrophages actually, are subsequent, that contribute subsequently to disease. And one thing that's central to the way that the plaque develops is this balance of pro-atherogenic and pro-reparative, or pro-inflammatory and anti-inflammatory macrophages. Traditionally, we tend to think of them as M1 and M2, but they're far more complex than that. But most importantly, the fact that a macrophage is present in the plaque isn't always a bad thing. We can get these reparative macrophages as well. So, having an understanding with respect to aging in particular, the impact of aging on the development of macrophages and their ability to become an M1 and an M2 macrophage is really important to understanding how that then subsequently leads to the atherosclerotic plaque. And Leah in the lab has been thinking about this in the context of the mitochondria. And the mitochondria in a cell are normally drawn as bean shapes that are just sort of these simple static organelles inside a cell. In fact, they're far more dynamic than that. Mitochondria divide and fuse, or fizz and fuse, all the time, pretty continuously. They form networks, and this controls the mitochondria ability to produce, to produce energy, to produce ATP. It also helps mitochondria with stress and damage control. So if there's any damage to any of its components, it can easily repair by dividing or fusing together. So the control of mitochondria fission and fusion is really interesting and really intricate. There's many different proteins and factors involved. And I won't get into that. But what Leah's noticed is that macrophages that have undergone this M1 or M2 polarization in culture have vastly different lengths compared to just a resting macrophage, which is denoted as M0. And this is true in both mouse and human. So she's seen that the pro-inflammatory macrophage is a much longer network of mitochondria than the resting or the M2, the non-inflammatory counterparts. And she can look at this under the electron microscope as well. You can see here, there are those beans, and then in an M1 macrophage, you can see kind of more of these really, really long networks of mitochondria. And if she alters the mitochondria ability to fizz and fuse, so if she knocks down all the fusion machinery or she knocks down all the fission machinery, she sees that there's a dramatic impact on the mitochondria length. You can see here, when you knock down fusion, the mitochondria can no longer fuse together. We have this phenotype that we call dusty mitos, because there's just these little dots everywhere, and the length here is altered. When you do that, you see a reduction in the amount of IL-1 beta that these macrophages can actually produce, and this is at the transcript level. Interestingly, if you do the opposite, where you prevent the mitochondria from being able to fizz or divide, so you're forcing them to have this very elongated mitochondrial network, and you can see here, you similarly have an inability for the macrophages to have to produce IL-1 beta at the transcript level. So there's something about the way that the mitochondria need to be able to come to fuse together and break apart in order for a macrophage to fully activate the expression of IL-1 beta. So what we think is happening is that, and again, we're still investigating this in terms of the mechanism, but at the mitochondria fission and fusion level, there's a communication to the nucleus. And in this case, we see it similarly in a macrophage treated with ox-LDL as a lipid-loaded sort of atherogenic macrophage. And we still see that there's, this elongated mitochondria network. And we think that that's controlling the macrophage ability to mount an inflammatory response, and that then therefore may control the ability for atherosclerosis resolution or the dampening of inflammation. And the way that this relates to aging, back to this clonal hematopoiesis, is that these macrophages and monocytes that come from these clonal hematopoietic, these risk factors, these mutations, have this sort of primed pro-inflammatory state. So they're able to produce more IL-1 beta and more IL-6. We hypothesize that there's something dysfunctional about the way that these mitochondria might actually be able to fizz and fuse in the setting of aging, compared to in a healthy normal setting. So just to wrap up, I hope I've convinced you that staring at the atherosclerotic plaque in its in its simplicity in terms of just thinking of it as a cholesterol blockage is oversimplified and certainly not. And now we know that inflammation is significantly contributing to it. But it's not just inflammation in the plaque that's important. It's actually inflammation elsewhere in the body. So the bone marrow and the spleen are major sites where immune cells are produced and regulated. And that has an effect downstream on just about everything related to cardiovascular disease. We focus primarily on the development of atherosclerosis in the vessel wall, but of course, a lot of these mechanisms could be at play in a similar way, following an MI in the injured myocardium. So we find that, factors like cell death, lipid regulation are, influencing the way that these cells are produced, and we're not fully sure yet how, but we think it's really the way that the immune cell environment is being regulated. We see that mitochondria, their signaling and potentially aging is also affecting the way that these macrophages are able to respond to inflammation, and potentially is going to be able to be help us understand why it is that in aging there's heightened inflammation and heightened regulation of these macrophages. When we see things like infection exacerbating our risk for cardiovascular events, it really makes us realize that we don't fully understand the interaction between, in this case, most of this is considered sterile inflammation, so it's not related to infection, and infection. So we need to understand that crosstalk, and something that potentially puts an individual at risk of cardiovascular disease, may be exacerbated by an infection or vice versa. And of course, one thing I'd be remiss if I didn't mention the brain-heart connection. So I didn't have time to talk about it, mostly because we haven't, we're just getting the project off the ground. But it's not surprising, not only that, of course, there's such a clinical connection between disease of the brain and disease of the heart, but mechanistically, a lot of what is happening in the coronary arteries, a lot of that pathology is mimicked in the vessels in the brain. And in fact, vascular cognitive impairment can be a result of either immune injury or a reduction in blood flow, similar to what we see in the myocardium. So we have a relatively new study that we've been fortunate to get support from a collaboration with some of our European collaborators, as well as some internal funding from the Brain Heart Oracle funding, looking at some of these mechanisms that I've described in the brain itself, and actually seeing whether or not the mechanisms that we understand in the endothelium, in the macrophages, are transferable to the understanding of the blood-brain barrier and the accumulation of inflammation in the brain during vascular cognitive impairment. And last but not least, something that I think we don't understand yet, but we're really connecting the dots a lot here. Your immune cells, your innate immune cells have a memory, and they're able to mount an inflammatory response and respond to an infection or a challenge. A significant amount of time can go by, and then they can actually mount a heightened inflammatory response based on some memory that they've had previously. Now, of course, in the case of your T cells and B cells, that's normal, right? So we were able to make memory B cells, which make antibodies, which help us fight off any subsequent infection. But in fact, your macrophages and monocytes and your natural killer cells and your innate immune system have similar memory responses that are not antibody-based, but are actually related to the way their nuclei are reprogrammed in response to the challenge. So that we think sometimes, I think that's going to underlie a lot of what we're investigating with regards to, whether it be infection, whether it be spleen versus bone marrow, and other, all the other various ways that our immune system is being primed and potentially exacerbating cardiovascular disease. So again, just ending off again where I began, thanking the group who did this. I didn't get a chance to talk about everyone's projects, of course, but we've got, again, a great group that has just been working so hard over the last couple of years in the pandemic. You can imagine how difficult that's been. And so I'm incredibly proud of them and all of their accomplishments, and I'm happy to take questions. Thank you.
Great, Katey. That's actually a real journey, that all the way goes from the days of the the microRNA, but then sort of the surprise finding MLKL and so I think it reminds me that quite often we are very heart or vascular centric, right? And then we realize, in fact, the diseases that we deal with the most commonly, in fact, is all systemic, and in fact, linked together, different organs and immune system being such a great example of actually linking the signaling, right? Amongst the different organs. You illustrate very nicely the bone marrow crosstalk with the spleen, with the the vascular, probably sort of other organs as well. Yeah, so it's really a fantastic example of that. And of course, we've been always interested in infection from some of the work that we are involved in earlier. And I always remember using the chlamydia, as a model of atherosclerosis, which activates innate immunity. And this is a little bit of kind of an interesting part that we may collaborate on, because one of my postdocs work on the MABs, that's mitochondrial antiviral protein, which is the crosstalk between innate immune system and the mitochondria, so there may be actually some interaction there, which is really interesting. Yeah. So
one thing I will say is that for years, people saw infection and they associated it with things like atherosclerosis, and they looked often for the bug in the plaque itself. And it wasn't really consistent, sometimes they would see it, sometimes they wouldn't know if it was like contamination, et cetera. And I think that there's, it was way before we appreciated the immune contribution to atherosclerosis development. So I think that's going to we're going to almost like everything, what's old is new again. And I think we're going to be able to go back to a lot of that primary literature and now understand it with the new, with these new mechanisms.
Yeah, absolutely. Yeah, especially in the post pandemic situation, and also the recent paper on MS, right? And the link with Epstein-Barr virus, figure that one out. Amazing. Yeah. So lots of questions. So maybe I just, certainly congratulations from Jim Robley, thought it was a very, very interesting and clear presentation, even to someone, who doesn't actually work in the field. Rob Beanlands has a whole series of bombard you with questions. Yes. And so amazing research. Thanks for your leadership. And actually multiple questions. So maybe we'll start out with the first one. And that's, so it's interesting that I guess, right now there's some suggested benefit of colchicine and the canakinumab. And, but then other anti-inflammatory medications like methotrexate and steroid, at least, in these clinical trials, don't seem to have a benefit, so is there, I guess, a clue there or any thoughts on that?
I think that, so colchicine, although not entirely known exactly its mechanism, but definitely canakinumab, they're really potent at reducing that sort of end stage of the production of the or action of the pro-inflammatory cytokines. Methotrexate steroids a little bit more nonspecific, so could have potentially more broad effects at reducing or incapacitating aspects of the immune system that shouldn't be incapacitated. And I think that's the next frontier is going to be figuring out precisely how to reduce innate immunity without compromising anything about the host, because with canakinumab, for example, none of the we won't see it as a therapy for chronic treatment of coronary disease, because for many reasons, but one of them being that there's a significant risk of lethal sepsis. So you really have to be careful to understand when you're taking away the ability of the innate immune system in particular, you may be really compromising host defense. So so I think that in some cases like colchicine or canakinumab, maybe it's the potency and the specificity of being able to really take away those cytokines. But again, we're still, that's just, that almost ended up being, those trials, just like, proof of concept from the pathway point of view. I think it really validated this idea that we've been observing for years and years, that adding an anti-inflammatory or some inflammatory component to a therapy could, in and above lipid lowering, could actually show benefit. So I think the principle is there, the specificity isn't yet. And that's why getting into the weeds of all of these specifics and the way they're activated, what's really interesting is there's a concept of potentially canakinumab benefiting people with clonal hematopoiesis, because you can genotype those people. You can take a blood sample, figure out, the proportion of their immune cells that have acquired these clonal mutations. And those immune cells have heightened production of IL-1 beta. So maybe the risk outweigh the benefit in individuals with clonal hematopoiesis. So that's kind of, we're talking about precision therapeutics a lot these days. That would be an example of where, for something like canakinumab, we might be able to use it in certain individuals. But we need to know more. We need to know way more about those really specific mechanisms in MI, atherosclerotic kind of environment.
Yeah, if I can just complement that, yeah, that's a great answer, and that's right on. And talking with Paul Ridker, sort of on ongoing basis, so yeah, so absolutely, yeah. So canakinumab, certainly for those who are able to actually lower IL-1, very specifically, actually get the maximum benefit. So it really actually shows specificity being important. And unfortunately, in the methotrexate and the steroid trials, in fact, those targets are not actually influenced, appropriately. And so. Yeah, so I think this really underscored understanding mechanism is really important. Okay, so here comes the other questions. Okay, Rob asked, so is that Dr. Tawakol, from Boston, a collaborator, and so says that there is a crosstalk of vascular bone marrow and amygdala in the brain. Of course, that's close to us, in terms of brain and heart. So it's an interesting question.
Yeah. So absolutely. I mean, stress response has so much crosstalk to the immune system. It's your immune system is really thought to be some of the ways that humans have have been able to evolve and live and survive is because of the way that we can respond to the environment, and it's primarily our immune system that responds. It affects, it'll then, go back to, our brain, it'll tell us to sleep. It'll, tell us, to whatever it's quite potent. So absolutely. And I think that's, ever since when you start thinking about the brain and you start thinking about its interaction with the immune system, what you realize too, is that there's very little that doesn't also involve the vasculature. So the vascularization in the brain, various, whether the immune cells are trafficking, or of course the blood-brain barrier is so critical. So all of those things also then just kind of come back around, and you realize that, what's in the brain and the circulation in the brain versus what is in the systemic circulation are so closely regulated. So I think that's going to become, all of those things. And I just see now here Rob's next question about MLKL in the brain. We don't know that yet. So a lot of these things are, as you can imagine, not being necessarily people who study and think about the brain all the time. It's usually like we see something in the vessel, we see something, maybe in the liver, in the gut or something. And then we say, okay, now what's happening in the brain? So, I think that tends to be our stepwise approach. But, I think that this is why some of these brain heart connecting, groups and it'll just, those links will happen a lot faster. We'll just make them faster. We'll make them quicker. We'll ask those questions a lot easier than we would if we didn't have these collaborations.
Yeah, so we got Rob to cue those questions, just to get people to think of the brain and heart. Yeah, thank you, Rob. And Tom has a great talk as well. And so the question is, in terms of whether reverse cholesterol transport, is inhibited in the innate immune signaling, and is that also playing part of the so it's really interesting. So again, like as I know Tom knows, and others probably know too, lipids like LDL and HDL can actually trap pathogens, and so can then part of the trafficking of the way that these lipid particles, normally endogenously is again part of the potentially immune system. So the way that they interact with the gut, the way that they interact with the liver. So there's a natural potential shutdown of things that then promote the trafficking of these lipids when infection or immunity is there. So it's entirely possible. I mean, what we just saw was just so dramatic, this idea that when MLKL wasn't able to traffic the lipids around the cell, I mean, could it be, and this is something that Adil's really interested in, could it be that there's, a cell has to almost make a decision? If it's infected, potentially it has to undergo cell death. But if it's, overloaded with lipids, maybe it has to traffic the lipids, and maybe it compromises. And how does it do that? And how does a single protein, participate in both of these things? And obviously, presumably the answer is there's many other proteins that it's talking to, and, all the different ways that it's modified and the layers. So I think, we're really, really interested in that, because, the same protein telling a cell to move around its lipids or undergo, dramatic cell death is just, is something kind of, we're thinking about it, and we're doing a lot of work on that now. Yeah, so I think it could be a threshold signaling effect, so in that it has a regulatory function, but when it exceeds certain level of threshold or convergence of factors, right, it switches over to a more kind of I guess systemic response.
And it's a good suggestion to look at reverse cholesterol transport in those mice too, because we could easily sort of link that to, and then look at, I mean, I don't know if anybody's really looked at the trafficking of lipoproteins to the spleen. I think we know that lipids go places and we know they need to go everywhere, and that's the way that lipoproteins, transport everything. But I mean, what happens to accumulation of, those bone marrow images where you saw this trafficking of lipid? Why is there so much lipid in the bone marrow? I mean, we know that there's adipocytes in the bone marrow, but why is there sort of lipoprotein in in endothelial cells if it is lipoprotein? So we'll lots of questions, I think.
Jim Robley has maybe just a quick, there are two more questions, so just answer them quickly, even though it's obviously complex question. That is that in terms of risk factor modification, like controlling blood pressure and statins lowering cholesterol, and what's their effect on inflammation?
Yeah, so studies have been done like that in mice in particular. So antihypertensive therapy does sort of reduce stress on the bone marrow, reduce activation of those bone marrow monocytes. So it's a similar idea. I mean, whether that translates to humans yet, I think is probably still under investigation in terms of, are the same mechanisms there, but certainly plausibly it could, especially that study I showed where individuals with either hypertension, atherosclerosis, or previous MI, their bone marrow cells are, activated. So you could then imagine that even if the therapeutic directly doesn't affect the bone marrow, just removing the stimulus of having, elevated blood pressure would then the same thing. Statins are really interesting because they have a direct anti-inflammatory benefit. Lots of different plausible mechanisms. One of them is actually part of this innate immune memory, this idea that it's actually part of the cholesterol synthesis pathway that the cells need in order to remember the way that they have seen a previous stress before and then be hyperactive. So it's possible that statins are affecting inflammation that way. Most people believe that the benefit from statins is really still just the majorly because of the LDL lowering, which is probably true on a population level. But mechanistically, statins have direct anti-inflammatory benefits. There's other things that cholesterol synthesis is needed for. So there's some thought about ways that, inhibiting cholesterol synthesis and things like that may not actually have a benefit. But I think we know overwhelmingly, the LDL cholesterol lowering outweighs any kind of, kind of minute mechanism.
Maybe the last quick question. So there's a question on the microparticles, so I guess from platelets and, interaction there.
Maybe notice a distinct lack of discussion about platelets or thrombi or anything related to thrombosis in the talk, primarily because it's incredibly complex to study at the mechanism level, at least in the models that we have. Our mouse models don't develop thrombus and things like that. You really have to artificially induce it, which is fine. I mean, our mice are just models as well. But that is, again, one of these frontiers that's really coming, is that, a thrombus is not just an acute event. It's actually related to direct crosstalk between microparticles, microvesicles, platelets, with immune cells. So it's not simply just an acute reaction that causes, the MI or something like that. So not something we study, but absolutely something that is, the way that the microparticles affect the endothelial cells, which will then, recruit immune cells, activate the plaque and expansion of the plaque is absolutely happening. So yeah, that's a great question.
Yeah, so it really actually shows the complexity of biology, and that we continue, with every discovery, there are more and more questions, and this is a great example. And but it's kind of a fun part, right? You can kind of follow the science, follow the passion, and you really have to, sort of be prepared for any component of the journey, and none of them is expected, right? And that's part of your title, right? And that's
almost nothing that we've done so far has been expected.
But that's fun, right? That's where you're really discovering new things, and it makes life really exciting. And this is how I think the success of your research, but also the exciting part of doing research together, right? You know, in which we can actually help to translate these. So this is a fantastic, great journey and a great illustration, you know, of how great science is conducted and the type of tools that we actually have available. So thank you so much and good luck on your, you know, continued journey, you know, and I look forward to more unexpected discoveries. And also great trainees in your lab as well. They're just fantastic. And so upcoming rounds. So February 21, no rounds is Family Day, and February 28th, Neil Vazdev, who's at the University of Toronto on new PET radio tracers for you know, imaging inflammation, you know, with a focus on neuroinflammation. Yeah, so maybe we'll find out amygdala is involved or not. And so, yeah, so that's the invitation by the trainee committee, who's always fantastic in doing a great job in hosting many of these sessions. So thank you so much for joining us and all the best, and enjoy the rest of the day. And thanks so much again, Katey. Take care. Bye-bye.
Bye.